All right, well, let's get started. Welcome everybody to Playground Global's Robochat series. For those of you not familiar, Playground Global is a venture fund based in Palo Alto, California. We invest in deep tech companies across 4 key domains: AI and automation, next-gen compute, platforms and infrastructure, and relevant to the discussion today, life sciences. And in particular, we look for life science companies at the intersection of biology, chemistry, and engineering and compute, and how we can apply modern engineering techniques to accelerate and enable the future of life sciences. The Robochat series itself is a roughly quarterly series of events where we bring together leading experts in the fields of robotics, AI, future of compute, to discuss and present cutting-edge ideas, trends, challenges, and what that future will look like. And at Playground, what we really look at is next multi-generational impact of the technologies that we work on. Before we dive into our topic for today, a few housekeeping items. This event is being recorded and will be shared with all of you following the event. If you have questions for the panelists, please submit your questions via the event Q&A feature on the right of your screen. We will take audience questions and also a few of the pre-submitted questions. And so please feel free to use the Q&A feature because we would like to add those to the discussion. The rough format will be about 40 minutes of panel discussion, followed by about 20 minutes of audience Q&A. If you are experiencing technical issues or would like to contact the event organizers, please use the event chat feature. All right, let's dive into the topic for today. Scientific discovery in academia, national institutes, global pharma and biotech companies brings life-changing benefits to humankind in the form of therapies and therapeutics. However, as a 2020 Accenture research report on the state of automation in life sciences states, most of the labs and the instruments within labs across the life sciences ecosystem remain disconnected, as Accenture says, serving as analog islands in digital oceans. But today we will not talk about computer-aided biology nor computational biology. We will actually talk about automation, how to automate scientific discovery and accelerate therapeutic development and related discoveries. All right, on to our guests. Nikhita Singh is the Chief Product Officer and co-founder at Artificial. I had the distinct fortune and pleasure back to like 2-3 years ago when Nikhita for the first time presented this idea of lab automation as the product focus for Artificial. And it's really amazing to see that evolve in now actual products and impact on the industry. I will let Nikhita now introduce herself and say more about her background, and then we'll get to Bob.
For sure. Thank you for the introduction and for having me here. As Sasha mentioned, I'm CPO and co-founder at Artificial, and we're building a software platform that makes it easier for labs to have scientific intent or experiments that they want to run and bring them to automated execution in the lab itself. My background before this, many years ago, was probably an overeager high school type A student who did a lot of science fairs. So I worked in a bunch of immunology labs and pesticide research labs, and got to sort of pipette on the bench and play with liquid nitrogen and all the fun things that I now get to look at from a very different perspective. And through that journey, I ended up meeting a professor who worked on surgical robotics and kind of got fascinated by the robotics world. And so worked on a bunch of things in surgical robots, in cloud media, and how you really think about robots as interactive tools in our day-to-day lives. From there, I ended up at the Media Lab at MIT. And so one of the things that was really interesting to me there is we were looking at robots that could be in unstructured environments like the home and aging, where things are messier, there's a lot of people, and it really conflicts with the rigidity of automation that you typically are used to. So as a personal passion, I don't like robots with eyes, and I also don't like them relegated behind safety fences. I like the hybrid space in between where you get to collaborate with them as tools. And so I'm pretty excited to do this talk with Bob because he's been an incredible thought partner for us in thinking about how we bring robots to the scientific lab, which I think is a really important purpose and full circle to my personal life story, I would say. So.
All right, to latch on your robots and in between space, Fun fact about Nikhita, she was actually in zero gravity with a social robot. Tell us about that one.
Yeah, so I think one of those adventures with the unstructured spaces was taking a fun social robot project into a zero gravity plane. So we got to run a couple experiments and have like a bolted box with 80/20 and kind of fly around. So it was a really fun adventure.
Nice. All right. Bob Gantzer, Director, Automation, Robotics, and Next Generation Sequencing at Beam Therapeutics. Tell us more about your background and interest, Bob.
Yes. So I am a molecular biologist by training. So to some degree, feel like a fraud in these conversations because I come at it very much from a scientific perspective, as you'll hear today, more so than a robotics perspective. But similar to Nikhita, I started out in the lab. I was actually a forensic scientist to start for a few years, doing everything at the bench by hand, then joined a few smaller startups, then a big multinational, and still doing everything at the bench by hand until kind of one day I saw the dusty robot over in the corner of the lab that wasn't being utilized properly and was, you know, ambitious enough or dumb enough to go over and try to get it to work and do my science for me. And sure enough, it worked and I was hooked. And so then for maybe the last 7 or 8 years, I've focused on how do we bring automation to scientists such as myself to elevate the field. So for the last 3 years or so, I've been leading that endeavor here at Beam Therapeutics, and then I oversee kind of our high throughput sequencing group also. So really my goal here is how do we bring automation to scientists with the lens of a scientist?
All right. Fun fact about you. You did automated seed testing in Brazil. Why in Brazil, and what was that like, you know, in that environment?
Yes, I was working for an agricultural biotech company at the time, and it ended up getting too difficult to bring seed and plants in and out of Brazil because of import-export constraints because of the Amazon there. So we actually went and built a lab and built and brought in automated testing and robotics into a rural area in Brazil. So that was a lot of fun, a lot of challenges in building in a farm, basically, robots that helped do seed testing. It was a great challenge. And I was backstage talking about how I still don't know any Portuguese, like spent a lot of time there, but still know very little. So that's still a shame.
All right. Yeah. Today I learned about the Northern and Southern Hemisphere seed regulations and how you maximize seed testing across 2 hemispheres. That's pretty cool.
Yeah,
all right.
Of course, when it's winter in Illinois, Iowa, then it's summer down in Brazil. And so you just travel down there and grow stuff in the Southern Hemisphere.
Yep. All right, so let's dive into the topic, automating scientific discovery, what Artificial is doing, what Beam is doing, and how to move the industry forward. So Nikhita, we'll start with you. Basically, we're going to look at the business drivers for automation in the scientific process. So the biopharmaceutical industry has its own version of Moore's Law. It's called Eroom's Law. Another thing I learned in researching, you know, this topic and, you know, all the stuff that I need to know to talk to you guys. Can you explain that, you know, and how it's kind of the inverse of Moore's Law that I'm way too familiar with?
Yeah, for sure. So I'm sure many of the people on the call are used to Moore's Law, which is really that computing power is getting faster and cheaper over time, exponentially. In the biopharma space, we actually see the reverse effect, right? So the idea is that it's getting more and more expensive for us to produce drugs and less efficient. And so, as you can imagine, that's obviously untenable for creating new therapies, creating new medicines, and bringing them out into the world. Back in the 1950s, for like $1 billion, you could ship 30 drugs into the world. And today, it takes anywhere from like $2 to $5 billion to get anything in the hands of real people. I think, you know, if you look at the space, there's a bunch of reasons why this is the case, right? Like there's the idea that maybe we've gotten all the low-hanging fruit, and so we have to be more intentional in the way we probe the biological domain. There's also the possibility that, you know, our efficiencies, both from a regulatory perspective and process perspective, add a ton of costs to the process over time. And there's also the point that, you know, we've been brute forcing a lot of the way that we approach biology with like high throughput screening or just looking at all the data and trying to see what sticks. And so I think the bigger sort of challenge here is like, how do we take the power of computation and automation so that we can change the way that we look for new materials, look for new medicines, and bring therapies into the world, and really leverage parts of Moore's Law to flatten the curve of Eroom's Law.
Nice. Hey, Bob, so a historical perspective would really be useful here. You know, in most industries, digital transformation and automation are basically table stakes at this point, and really not competitive advantage. You know, kind of to compete, you just need to be, you know, you have digital twins of your industry and your process and stuff like that. So why is not the case? Why is this not the case in labs in life sciences?
Yeah, I think there's a couple reasons. I think this image I really like. I take it from a white paper published by Antha. So thanks, Marcus, for letting me use this. But if you look at the automotive industry, as you said, in the 1930s, and you fast forward 80 years, it's all automated. It's completely revolutionized into a different way of manufacturing. But if you look back at Alexander Fleming doing some, you know, of his research in the 1940s, and then you look at labs today, they look the same. They're not wholly different. You still have a person there, and usually in the image in the 1940s, someone's got a cigarette and they're sitting there pipetting. And the only thing that's changed now is there's just nobody smoking in the lab. Other than that, it seems like it's almost identical. And I think the reason for that is maybe twofold. One, I think there's a cultural challenge in the sciences still in adopting some of the technology. I can kind of give an example of at Beam, the company I'm at now, I was one of the 30 first employees here, which is really unheard of to bring automation into an organization that early. Usually within biotech, you'll maybe hire a couple hundred people, throwing people at the problem before you start to automate it. And so I remember when I was doing my interview, we had a brilliant scientist that one of our founders that just halfway through had kind of asked me the question, like, I don't know why we need you. And she'd gotten, you know, infinitely far, was honestly one of the top scientists in the world, but still hadn't fully embraced the impact that automation could have on the sciences. And I think a lot of that goes back to, in academia, it's cheaper to hire an undergrad or have a grad student do it than to automate. You know, we don't end up teaching scientists programming. You're not really exposed to lab automation until you get to industry. So there's a big cultural hurdle there. And then there's also a technical hurdle. You know, Nikhita had mentioned the composition space of robotics, where in the car factory, like you see there, it's pretty easy. It's a manufacturing line. And I know it's not easy, but it's a manufacturing line where you're doing kind of the same thing over and over. But in lab automation and research, it's getting to be different. Scientists will have a problem that they'll come to you one day and say, you know, hey, I've got this idea. I want to run tens of thousands of samples across dose curves and generate hundreds of thousands of data points to see if X, Y, Z is true. And you say, okay, you know, how long will it take you to do it by hand? And it's like, oh, it'll be a week of painstaking work. I'm like, okay, well, will you ever do it again? It's like, I don't know. It depends what the answer is, right? If I get the answer that I think is interesting, then yeah, I'll want to do it all the time. If not, then no, it was a one and done and it wasn't a discovery and it doesn't matter. Well, that gets to be tough to automate, because I could turn around and say, okay, well, you could do it a week by hand, or I could spend a week programming it on the computer. You're probably just going to have to do it by hand. And so we still have this technical challenge of automating laboratories in a way that allows you to do bespoke experimentation. And there's still a big technical gap there too.
So did the scientists that asked you, why are you here, change their mind?
Yes. Yeah. So she did. I did get hired. Now, there's a funny story. Part of the conditions of my hiring is that she gets to name all of our robots. And so you might see a picture of one of ours in a slide coming up later, and the robot's name is Ursula. And so it's named Ursula because all of our robots at Beam are named after Disney villains. And so if that doesn't tell you about the perception of automation or robotics in the sciences, that they're all still viewed as villains, I don't know what else will. So I think it just drives home that there is kind of a cultural aspect of automation adoption in the scientists that we really, or in the sciences, that we really need to overcome.
All right. Hey, Nikhita, next question to you. Basically, why is this an exciting field? You researched the entire life sciences ecosystem. What were some of your key findings that made you decide to direct your passion, energy, and creativity towards this space as opposed to everything else you could have done? You could have been now flying in space doing, you know, very different stuff, but you chose this as your life's work at this point.
Yeah, I think one of the things like on a personal level that really stuck out to me was, coming back to the space and visiting all these different labs, like 15 years after I'd been in one, I kind of expected the nature of lab work to look very different and for me to sort of feel like a total noob in the space. I definitely felt like a total noob in the space, but I think the nature of lab work stayed pretty similar to a lot of what I was doing back in the day. Like there was still pipetting on the bench, there was still notebooks and handwritten Post-its that I saw like put on liquid handlers and different devices. And it's not to say that, we haven't had incredible advances, like the boxes that we have as machines in the lab are far better, like we can sequence faster and cheaper, we have better technologies. But I think a lot of the nature of the work itself has stayed the same, right? The way a scientist spends their day to day. So for me, like one of the big parts was just that jarring kind of perspective, like walking into all of these labs and seeing that there is a clear need and desire to change the way that work actually happens. The other part, I think, for me, like there's a need, but you need to also have a desire from the folks who you're designing for and building for. And one of the things that I always thought was really fascinating was that out of like the 100 or so customers we probably talked to in Artificial's journey, they have a robotic automation journey that looks something like this, right? Where when a company starts up and is doing their initial R&D, they pretty much have everything as manual. Like as Bob said, you hire the student and you kind of make them pipette and do all the work that you need to do. And then slowly over time, you start to integrate a couple instruments, like maybe you buy a liquid handler or start to build out automated processes. And then over time, you end up in the more fully integrated world. And so if you look at like folks like Zymergen and Ginkgo who've been in the news and IPOing recently, they've kind of taken this spectrum and gone all the way, right? Like they have far more factories. They have this notion of foundries with like lots and lots of automation that they're driving towards. And there is really a testament to the value of automation when it comes to repeatability and being able to get data at scale that they've shown. But not every lab has the access to be able to do that, right? Like as Bob said, he was an anomaly in being hired so early. And I think that's a story for many, many companies where they don't even know where to start with automation, let alone where they want to take it. And so there's obviously the core need in the life sciences space as a whole, but I think there's also an emerging desire in order to invest and look at automation as a tool for the life sciences space. And I think the final part that stuck out to me was like, as a product person, I love the user side and like thinking about how you can change the nature of work itself. Automation and robots sometimes get a bad rep for like replacing jobs and like all that. But I think something that's interesting in this space is that lab automation engineers are like a totally new role, right? I've met people who are ex-biologists who are like worked on rockets before or physics majors that like randomly ended up in this space. And I think the idea that you can shape a role and create a new type of job market for people to invest their energy in is like exciting to me. And there's yet to be like a Stack Overflow for the life science and lab automation space. So I think that's like a really exciting opportunity. So I think in summary, it's really like there is a need, there's a desire, and I think there's a capacity to change the way that we do work. So.
Yeah, that last one in particular, I'd like you to elaborate. So why would an aspiring AI roboticist, that can work on self-driving cars or rockets or who knows what, actually come here and, what impact can they have with their skills, but not necessarily know anything about biology?
Yeah, I mean, so there's the obvious answer, right? Of like, you have clear impact into the world. And I think that's something that resonates with a lot of folks, right? And I think it's particularly relevant given the year we've all had. I think on a nerdy sort of intellectual level too, right? I think one of the things that's really hard about unstructured environments is knowing the scope and being able to know your physical world in a concrete way. And so as a roboticist, it's a luxury to know that plates are always the same dimensions. And, there's like a standard set of formats for all the things that exist in a lab. And so as far as like the unstructured environment with enough structure that you can actually like, do this work, I think it's a really compelling playground for investing in like new ways to leverage robots. So, both from a nerdy perspective and also like the social cause perspective, I would say it's a great place to work.
Bob, any trouble attracting those nerdy people to work on this stuff?
Yes, I almost interrupted and just said, yes, if you are those people, beamtx.com. We've got plenty of jobs posted, or just please join us in the industry, because a lot of folks in my role end up coming from the sciences. We end up being those frustrated scientists that are tired of living at the bench and doing things manually. But what we lack then is some of the technical expertise. There's not enough software engineers doing what I do, so I'm probably not doing it right, and could benefit to learn from some of the tech, the technical side that people could bring to it. So it's a burgeoning area with plenty of opportunities. So if this at all interests you, one, please feel free to reach out or check out our website, but explore it more. It's a lot of fun.
Well, okay, along those lines, so what is the current state of the art? Somebody entering the industry, what are they faced with? And give us an idea of the technologies and where they are with respect to other industries, with similar technologies.
Yeah, so I guess going back to the spectrum that Nikhita showed there, the manual to semi-automated, this is kind of what that looks like. So here's a Hamilton Vantage. This is when we're talking lab automation. This is the workhorse of lab automation. It's what's called an automated liquid handler. And essentially it is doing the pipetting that we've kind of talked about. It's finally something that's automating the pipetting of liquids. So you can see two of my engineers there working. It's pipetting out of that plate and just moving liquids around. So that gets you part of the way there. But then kind of as you go through the next step, and what we're starting to do at Beam and what folks in the industry are starting to do is tie those together. So integrate that a little bit. So in the middle of the screen there, you see a precise arm, a cobot on a rail, and now it's starting to connect to a lot of other devices. So when you're thinking about scientific experimentation, it's kind of like cooking. It's kind of like a recipe to some degree, but maybe a lot more complicated, where you're just adding liquids, you're incubating, so you're just cooking it for a certain amount of time. You're adding more liquids, you're spinning it down, you're peeling it, you're sealing it. So a lot of things there that you have to do to execute an experiment. And so now companies are starting to automate all of that. Instead of just a piece of the journey, you're able to automate all of that. The fun thing gets to be is now not only do you start to get more walkaway time where you can just kind of load samples on and come back to the end result of whatever you're looking for, but what gets to be even trickier is automating all the data that then generates. And so I think that's kind of where we're at now, but then hopefully are going to start to get a lot better at that in the future too, as looking at lab automation and the data of lab automation kind of working more hand in glove together.
Hey, actually, the data collections interests me. What's the volume of that data? Are you talking really terabytes, petabytes, or is it still much smaller scale?
So yeah, you can be talking terabytes, particularly as you're automating. There's companies out there. So when you're starting to do sequencing, you can get into that realm where if you're looking at, what is it? I'm going to get this number wrong, but 3.4 base pairs in each genome or something like that you're sequencing. It might be the other way around, or then even as imaging. So now there's a lot of companies out there that you'll be actually like imaging cells to look for morphology changes in the cells as an indication that a therapy might work on a particular cell type to elicit whatever effect you're looking at. And so you can imagine that if you're imaging, typically in those plates that Nikhita referenced, you can have 1,536 wells or 3,456 wells per plate, and then go through hundreds of those plates, generating hundreds of images per well. The data really starts to add up. And that's, again, where automating that becomes really important, because if you're generating that much data, you got to make sure you're automating the data capture of that too, not just the execution of the experimentation.
All right, that's really cool to know. So to dig even deeper now, Nikhita, question for you. So why is this so hard to do from a robotics automation perspective?
Yeah, so I mean, if you go into a lab today, right, like even when we were talking to Bob at Beam or folks in other parts of the automation journey, even if you go all the way to the fully automated world, there's still operators and scientists who are putting stuff in and out of systems. And they often, will take it out of a system and go do things on the bench and then bring it back into the automated system. And I think that has a lot to do with the fact that scientific discovery is still like an art form. And I believe it'll continue to be and should, and inherently an iterative process. So that means that when you have that confront the rigidity of robots and automation, where you need to have these hybrid workflows, it becomes becomes more complex, right? So you need to support, manual alongside automated. So that's sort of one chunk of it. I think what that introduces is this weird 3-body problem where you have the human, you have the robots and automation bit, and then you have the software that they're using to interact with their world. And when you want to reconcile state between these 3 things, it becomes really difficult. Like as a scientist, if I go rogue and take a plate somewhere and put it somewhere else, how do I tell my robot I did that? Or how do I tell my software system I did that? And so it becomes this fundamental UX challenge and technology challenge to have state reconciliation across all of these tools that you're using in the lab space. And I think the other part that also makes it difficult, like to Bob's point about, Ursula and having all the devices that people are putting together, I think a lot of lab automation grew out of needs. And so people built a smarter and better device or box that could do X, like plate seal really well or plate read really well. But they didn't really think about how those all stitch together. And so you have these software and technology silos that are great on their own, but there is no orchestration or overall experience that brings everything into like an easy to use layer. So to me, those are kind of the 3 big chunks that make it hard from a technology and robotics perspective.
Yeah, so it's basically a workflow. You go from one station to the next, and I bet exception handling is particularly hard.
Yep.
Not just human, going rogue like you said, but even robots make mistakes.
Exactly. And so like, I've heard sort of funny stories of like someone coming in and the plate was flung across the room and it looked like kind of a, just like a random plate murder scene in a lab and things like that. And so, at the end of the day, like it's really difficult to manage all the exceptions introduced by both the people and the robots when you're building software.
Yeah. Right. Well, Bob, why is it so hard from the scientific perspective?
I think it goes to Nikhita's, the first thing she said is the artfulness of science, right? Like there is an art form to it. And so when you go to automate that gets to be very difficult. I mean, when it's on paper as an SOP or a recipe, it looks easy. And typically you can program that up in an afternoon to repeat the tasks, but it's the nuances of what the scientist is doing to get that experimentation to work. Particularly if you're working with cells, for example, that are living organisms, they have to be treated as such. That you have to have kids' gloves, and there's a bit of skill that needs to be honed over years to get them to behave in a way that you want them to. And so to automate the art gets to be really difficult. But on the other hand, I think it gets to be a necessity. Nikhita earlier also had mentioned there's a reproducibility problem in science right now. There have been papers that have come out recently just showing that up to 40% of peer-reviewed published results have been difficult or impossible to reproduce. And so I think some of that goes to the artfulness of it, right? Like it's hard to put down in a peer-reviewed paper the SOP that you did of how you dispensed your liquid on the side of the wells so it didn't disrupt your cells versus just squirting it right on top. And so if you write that into code, though, it allows those protocols to be actually replicated in a way that should be much more meaningful to scientists. So it's challenging, but again, continues to be a challenge worth rising to the occasion for.
But along those lines, does automation, how does it impact the visual senses of a scientist, or the intuition they have when looking at a petri dish or something? And if you automate it, and you automated something for 2 minutes, but it really needed 2.2 minutes and 3 seconds, how do you deal with that?
Yeah, that continues to be a challenge. So, I mean, you're seeing more and more vision systems being integrated into platforms to, say, record, cell densities at dispensing. But again, there's the flip side of it. There's a term called confluence when you look at cells, which is basically like how much of the cell layer covers the bottom of the plate, and it's measured in percentage. And so if you go down to our cell culture lab here and you have somebody pull out a plate, they might say, oh, this is at 70% confluence, because it's a complete eyeball guess, right? Like it's, and so then if you go to hand that method off to someone else and you say 70% confluence, your 70% might be completely different than someone else's. So part of the technology that's being developed is coming up with software and hardware that can help to digitize some of those measurements to make it so that we can start to translate the art into code.
All right. Well, now with all this background, historical perspective, current state of the art, how did COVID impact life sciences automation? Has there been acceleration like in other industries? You know, what's the current thinking?
Yeah, I can take that one. Without a doubt, there has been. So I think there's been more of those liquid handlers that we showed the Hamilton sold in 2019 than in any year ever. And that goes for like every liquid handling company across the board. So there's going to be a glut of lab automation out in labs like we've never seen before. And so when COVID testing hopefully starts to die down or changes how we do it, there's going to be scientists now that have been exposed to these technologies that'll then start to, you know, change it to whatever science they want to work on next. So I think that's going to be great. And I think the other impact has been the organizations that adopted lab automation early and that had the infrastructure in place weathered 2019 and 2020 and 2021 much better than others. I know here at Beam, the group that I lead in the sequencing core, we track our throughput on a weekly basis, and it didn't dip at all. Throughout all of 2020, we were able to maintain because we were able to rely on the automation now to be executed by one scientist safely in an isolated room. Whereas had we been a company that had just thrown people at the problem where we needed 15 scientists to do the work we're doing, we would have been shut down. We could not have done that safely throughout the pandemic in the same way we were able to throughout. So I think we were really fortunate to be one of the companies that really adopted it early and had the infrastructure in place for COVID.
All right.
Just to add to that too, I think one of the things that we found talking to some of our customers too is like people are changing the way that they collaborate on work in the lab. So like, you know, typically you would have scientists next to each other showing how to do a particular experiment or a process. And now people have to spend time on Zoom or Slack in order to transmit that same information. And so I think there's sort of this willingness to rethink what it means to collaborate in the biological space. And I think that's kind of an interesting opportunity to design tools for this new way of work.
So let's turn to the future. The really fun part, the vision, you know. So starting with you, Nikhita, if you could paint that vision, you know, what does it look like? Is an operating system for scientific discovery within reach, or is it a fantasy? And, you know, look as far ahead as you want.
Yeah, I mean, so I think one of the things that's kind of interesting, right, is if you want to engineer biology and make it more reproducible, you need to have a certain level of control over it. And so I think that lends itself to this question of like, what is the set of applications that helps us have more control over the work that we do? And for me, I think I want to get to a world where every scientist is empowered to leverage robots and automation as a tool in their day-to-day life, right? And I think there's a massive opportunity to kind of rethink that space, both from a cost and efficiency standpoint. So like, how do you bring automation infrastructure and make it easier to implement and empower the set of lab automation engineers who are doing that work? And then there's the part about, I want a future where a scientist walks up to an automated system and is not immediately intimidated, right? Like if I had a work cell show up at my door, I personally would not know what to do with it. And so how do you create that out of the box experience that you can get in all sorts of other tools in your life, so that someone can just get up and running with automation much faster? I think the other part is really the access bit and like creating a future where it's not, automation isn't just for the Zymergen and the Ginkgos of the world with like massive amounts of capital, but accessible to the R&D lab that's just starting to think about it. And I think to make that a little bit more tactical, you know, some of the ideas that we think about at Artificial is really around tools. So we have an editor that lets you actually describe the physical environment in your lab. So in this case, a scientist could go in and actually start to describe and build out the lab that they have in their space. And we've encoded all this equipment from different vendors that you saw in Bob's work cell, for example, with common sense lab physics of like what can hold what types of plates, what is compatible where can things go? And so all of this information that was previously tribal knowledge that lives in the brains of different people is now being encoded in editors that people can use to quickly collaborate and build out labs in their space. The other bit to our conversation about, you know, the artfulness of science and encoding scientific intent, we really think a lot about how do you take natural language and easy tooling so you can start to digitally encode these protocols. And so can we have a future where protocols are versioned and they're articulated and you can see how they've transitioned over time? And so support the iterative kind of nature of science while still digitally cocooning it so that you have traceability, you have reproducibility in a meaningful way. And so this is one of our editors where you can start to write out the protocols that you have, refer to the physical world, like where is this Hamilton, what am I touching, and guide people through that process. And finally, I think, for me, I've always envisioned a future where people and machines work together. And so I think this gets to your operating system question of like, I think about operating systems like your Windows or Mac or your phone OS. They're not inherently useful if they're just sitting on your desk. I think you need a person to go pick it up and do something with it and like get some benefit. And so in that regard, I absolutely believe like an operating system is required and possible for scientific discovery, but it will always have people in the mix driving what they want to do with that work. And so this is like an example of a workflow similar to what we have at Beam, actually, where you have this hybrid world of people doing things on an automated system, and then robots actually running and executing work, and then people interacting with it yet again. And so I think if you can create like an operating system that supports both of these realities, you can really go pretty far. So that's what I hope to create in the future. And I hope artificial gets to play a part in that to whatever extent we can.
So your vision still is with an expert in the loop, even in the distant future.
I think so. I mean, for me, like I've never been a proponent of like, here's the black box, then you shove your desire in and you get the therapy out. But I think that you can make that box smarter and better and like more usable to scientists and folks.
All right, Bob, what's your vision?
Yeah, what gets me excited as I look, so shorter term, just augmenting in the same way that Nikhita had mentioned. So using these automated tools in the same way that you're using pipettes and centrifuges and really having scientists comfortable. So everything that Nikhita had just talked about. And then taking it, she had mentioned the access side of things. And that's one thing I'm really excited about in the future is, again, the Ginkgos, I'm always jealous of all the tools that they have there. And we've got some pretty nice setups here at Beam, and the big pharmas have their own too. But it's still not in academic labs where a lot of the base research is being done that enables all of the technology that we're using. It's not accessible in smaller startups that need it. And then it's not even accessible to folks. I grew up in the Midwest, in the middle of nowhere. And so if you're not on the coast, you're never going to see the amount of labs that say we have here in Cambridge or that are out there in San Francisco. And so I think a world in the future where there's a concept of cloud labs that Nikhita and I have talked about a little bit, and they exist and they're morphing still now, but where somebody from anywhere can just have digital access to the physical labs. I mean, there's not really a reason why we all need to be located here in Kendall or Central Square of Cambridge or in San Francisco. Eventually, there should become a future where ideas are what matter, and having creativity that really is what you see in the most brilliant scientists that work here in Beam is like, that's the specialty that makes their discoveries so interesting, is the creativity that they bring to the art of science. And so I think using automation to make that more accessible to people is just going to benefit humanity in the amount of discovery that we get.
Yeah, that's a common theme with automation and AI in general, that it democratizes access to the tools and to the science, and now you can do it from anywhere, especially with a cloud lab. All right. Well, I'm going to turn to some audience questions now, both pre-submitted and live questions. And I really want to start with an awesome name that I recognize, Gabe Warshauer Baker, that I had the awesome pleasure and fortune to work with at Cruise. Gabe, good to see you here. And he asked a similar question as Carlton Amu, so I'm going to combine the two. But basically it has to do with, how does machine learning come into the picture? Now that automation lets you generate such a large volume of data and to analyze it. And the way Carlton said it, what are some other use cases for ML are you finding to be meaningful? Let's start with you, Bob.
Yeah, I think AI and ML in research are finally having their day where I think we're going to start to see some discovery coming from it. And the companies that are doing it well are the ones that already have the lab automation infrastructure in place to make sure that the data they're generated is curated in a way that you can actually do something with it from an AI ML perspective. So that come to mind, Recursion is a company that I follow closely and really respect a lot. And one of their core concepts is they're actually taking what you might call recycled drugs. So drugs are small molecules that have been overlooked or just weren't thoroughly discovered enough to find all the applications accessible to them. And so what they're doing is they're throwing a lot of automation at it to test these drugs at scale, and then using AI and vision systems of cells, similar to what I was talking about before, to then find those discoveries. So they're actually able to go back into the haystack and find more needles than we even realized were there, just because of AI and lab automation.
Nikhita, along those lines, is there, I liked your digital twin simulation environment or lab replication environment, but, we talked about exceptions and things that go wrong. Is there a concept of actually learning and training the lab to get better and learn from exceptions, or is that still too far-fetched?
Absolutely. I mean, I think one of the advantages that you have when you think about the blended work of like people and robots is you can learn what exceptions happen commonly and then start to build out tools to reconcile them, right? Like whether that's a robot that can learn to reinitialize itself when a certain set of exceptions occur, or it requires someone to come in and reset and like put some plates in a new place. Those are all potentials, I think, for process optimization where ML can fit in. And so one of the things we spend a lot of time thinking about is like, what is the set of algorithms that's orchestrating the lab itself? And how can I learn more and more about how long certain tasks take and how I might batch experiments together better, or how I might operationalize and optimize the way that I run my lab from an efficiency standpoint. And so that's like an area for us that we've invested a lot of energy and time into, is like sort of creating that orchestration engine and logic for running lab operations.
All right, so Alan Miller has a question kind of back to the early, part of our discussion about the history and how we got here. He basically is asking, are the automated processes more reliable and repeatable than the grad student implementations, at least enough to make them more attractive for that reason?
So if done right, yes. I don't think that there's much doubt there from my perspective. I just remember being in grad school and coming into the lab and being tired from the night before, let's say, and then not performing optimally when like you were up late studying or out with friends. And so, your consistency with automation is really something, worth having. But I think more importantly, it's the extensibility of it, where you can then export that method and then share it and have it executed the same way across a number of different platforms too. So it's the way that what we find the most benefit from is when you're sitting there pipetting and executing a task, you're moving a sample from like A1 to B1, and then you kind of space out and you think to yourself, you're like, oh, did I do that? Did I put it in the right place? Did I actually add this? And then you talk yourself into yes or no that happened, and then you look at your results and you assume that it did or didn't happen. Whereas again, if it's automated, it's all of it is tracked. So you've got logs to show exactly what happened. And so if there is an error, if there is something that might have led to a lack of reliability or repeatability, then it's there in the logs and you've got the opportunity to fix it. Whereas if it was a person doing it, unless you've got like a camera in the lab or something that you're going to go back and look at, you've really got no way of telling exactly what happened in that experimentation.
Hey, Nikhita, so, Artificial obviously builds a product for automation and has to sell the value proposition to companies like Beam and others. So are there some metrics that you actually have in place to actually prove or demonstrate the value add in terms of repeatability, throughput, whatever that metric may be?
Yeah, it's an interesting question. So I think for us, like in our journey, classically automation always has this like ROI calculator that comes with every investment, right? And I think the part that's been really fascinating for us is talking to all these labs. They come to us more with the accessibility challenge of being like, I'm not even at a point where I want to do the metrics perfectly. I just want to make it used more and more often. Mm-hmm. And I think part of the gripes that people often have is like, if my scientist doesn't use this automated system to run the experiment, they're going to go do it on the bench instead. And so I've lost traceability. I've lost like a certain amount of access to that information. So I think, one is like the accessibility bit is usually what we've seen draws people in, despite like our journey through the ROI calculator space. And then I think there's like more advanced labs that have many systems that want to look at how they can increase throughput and how they can maximize the efficiency of the work. And so things like machine utilization, right? Like how much downtime is there? How much is something being used? And can you scale that metric? Can you actually coordinate the manual steps for operators that need to happen between stations so you're not losing as much money on just operational inefficiencies? Or I think there's a big kind of need for clinical operators in the space because there's massive turnover at many companies. Like you lose about 30% of people per year at some companies from what we've seen. And so how do you train up people faster to use these advanced systems and manage some of that turnover challenge that companies face? And so I think those type of bits have really been resonant with folks of like training costs, optimization across these hybrid workflows, and then being able to increase the utilization of the devices that they've invested in.
All right. So earlier we talked about COVID impact and how we handle that. So there's a question from Shen Wang that's kind of relevant to that topic. How are the backlogs of the relevant equipment changing with the pandemic easing? I guess that's a question about the supply chain and the availability of the equipment now that we have to deploy. Bob.
Yeah, it was bad there for a while. And thankfully, we're starting to see light at the end of the tunnel. So for the Hamilton, again, that we've shown, that I've mentioned is kind of the workhorse in the lab, it uses disposable plastic pipette tips for every transfer so that you're not contaminating your next sample. And we placed I don't know if I want to make the company feel bad about that, but it took months, like 6 to 8 months to get that back into supply because, as I mentioned, those devices were just being launched everywhere. And rightfully so, those companies were prioritizing COVID research over maybe what we were doing because we were in the middle of the pandemic. And so we had to start adopting like tip washing protocols and spend a lot of time automating that process and validating that process that would actually work. But thankfully, we're starting to see a light at the end of the tunnel a little bit where supplies are starting to trickle back in. Lead times for devices. I mean, that's one of the things that still is very challenging in the lab automation space is if you go down this journey and you say, I've got this process that we're going to do a lot at Beam, let's go ahead and automate it. I think idea to like execution now is still maybe on average, I would say about 18 months. So it's still a really long time. A lot of that is just lead time at vendors to get some of these supplies in, design time, implementation time. So it's still not a very quick implementation, but thankfully COVID is easing a bit and there's a little light at the end of the tunnel. But it was very difficult there for a while.
Nikhita, are you noticing anything as a solution provider? Does that backlog affect you at all?
I think the bigger thing we've noticed is actually like more people are resonant with the investment in automation ever since like this journey. Because I think the way that lab work was being done fundamentally shifted, right? So there's a lot more push for like, let me buy all the liquid handlers that I can get my hands on. I think the other part has really been the fact that, as you're getting more of these integrated systems, like we've been working with Thermo on an amplitude system that they have for COVID testing. And so now that you're like shipping these out at an unprecedented rate and trying to get like software and hardware out there, how do you make it more and more accessible to the end user and train up people quickly? And so I think from that perspective, it's put an emphasis on the investment in automation in a way that we hadn't seen like a year and a half ago, I would say.
Yeah, this brings up in my mind a thought about, you're talking about assembling all these different equipment, different robots, different handlers. There's a lot of software, firmware, drivers, that needs to be aligned, and there's some scripting language on top of all that. Can you talk about that can of worms? How do you deal with that diversity of version control in general, with all this equipment and the diversity of the robots that you have to deal with?
That is a challenge. It's the challenge. So most of the time, there's few shops out there that build a lot of these themselves. So like from Beam, for example, we partner with High-Res Biosolutions on a lot of the systems that we've made public, and then they partner with a lot of the individual device providers to then kind of build these systems. So it's through kind of a layered partnership of hardware, and then the granularity of the software by the time it gets to us is more the execution of the automation to like to make the science happen. So it's, you know, it's the last mile problem is kind of what we face. I'm not designing the cobot by any means. I'm getting the cobot, somebody's putting it in for me, and then I'm making it do what we want it to do. But there's still a lack of modern software technology that's embedded into lab automation. Again, it kind of goes back to the first picture we showed where we're still like 30 years behind in some respects, where some of the softwares we work with don't have version controls. So if you go onto our PC in the lab, you're going to see underscore version 1, underscore version 1 really, underscore version 2.3.7, underscore this is the final. Like those kind of things unfortunately are still happening. And so you have to put like physical best practices in place within our organization to manage that, because technically that's still an opportunity. And it's something where like version control is one of the first things we came to Artificial with, and that they're kind of helping us in that regard, to where when we can go to Artificial and just go to one spot, we can send our scientists to their platform to say, I want to run this assay on these devices, and it's going to make that connection for us to the right version of the software that we want them to run, so that they're not going to have to go into the Hamilton and pick DNA isolation version 2 underscore final Bob's method. Like a scientist doesn't have to look through all those logs now. And that's one of the things that the team at Artificial has really helped us with, is to incorporate version control into the process.
Yeah, Nikhita, like Artificial platform is supposed to abstract all this complexity from managing it. Basically, you manage the fleet of the diverse robots. So what is the state of that now? Is it good enough? Still a lot of work to do?
Yeah, I mean, I think there is, you know, as Bob said, it's a very layered problem, right? There's like the level at which you can drive the equipment and have control over it. And then there's a level at which you can design on top of that equipment to do these larger workflows. And so I think we've taken very much like a top-down perspective of like, how do we unblock the intent at the top and then slowly move our way down into more control and active participation in the devices themselves. I think, you know, the good thing is like a lot of the vendors these days are actually pretty excited to share things like models and start to like version some of these things, because I think there's been a push from folks on the customer side to be like, can you version your drivers and, you know, have the state of the world there. So I think we've taken very much like a start at the top, partner with folks like Hi-Res and people who are building this equipment, and start to sort of catalyze more of an ecosystem view on how we kind of unblock this future together. So.
And if I can add to that, it's finally the adoption of APIs in lab automation is becoming a thing. Like again, Hi-Res is leading in that, where they're just making their scheduler, which orchestrates a lot of those, accessible to devices like Artificial, which makes the whole ecosystem work. A lot of the new liquid handlers that are coming out are now being a lot more API accessible and friendly. So some of the challenges that were there from the technical side are finally starting to be lifted, as kind of this modern-ish wave of software is finally getting adopted in lab automation.
All right. Hey, Nikhita, a question from you from the audience, from Patrick Astarita. How can we promote cloud lab cost competitiveness?
Yeah, it's a question we've thought a lot about. And I think, you know, Bob mentioned him and I have noodled a lot on this subject as well. I think one of the things that's been hard with cloud labs, right, is you have to give up a certain degree of control over the experiment that you have. And so I think cloud labs historically have been great at supporting like a base set of experiments, but then when you want to do anything off-road, then that same cloud lab doesn't support it, and you're stuck with this decision of like, well, I still have to buy this equipment to do this experiment that I want. And so I think part of this has to do with like the trends we were talking about earlier in our conversation, where as we have more control and agency over our biology through engineering the biological domain itself, we can be more precise in what a cloud lab enables for us, right? And so I can set up a cloud lab with a focused purpose that, you know, anyone anywhere can then access for that type of experimentation. So I think driving towards more verticalized sort of core solutions is like one way that we can move towards that trend over time. I do think at a certain level, like labs will still need to exist and have their own R&D in their own environment, and then you can outsource and distribute some of the work that you want to do and kind of have this more hybrid worldview that you're enabling. So I hope that we can get there, especially with this like push towards more computational and engineered biology that's taking place.
Bob, are cloud labs even part of your workflow today?
They're starting to be. Yeah, we've actually been talking to some to augment. Like I said, there's an 18-month lead time on anything new we want to do. So some of the benefits of the cloud labs as they are now is they allow you to prototype and even just try devices that we don't have to buy, that we can maybe prototype and use, see the validation and make sure the science works in an automated fashion before we dive in and make a big investment in both time and money in bringing something in-house. I also think that there's a shift in biotech and medicine more towards cell and gene therapy, more towards molecular biology instead of traditional small molecule chemistry. And as a scientist that's automated the various forms of science, it's much easier to do molecular biology. Everything's water is a buffer, it's aqueous, it's not as volatile, it doesn't need as much controls. So I think actually the transition in medicines towards molecular biology is actually going to help facilitate the adoption of more and more lab automation. It was going to happen anyway, but I think it's going to make it even easier to do some of the experimentation that we want to do. And so some of these cloud labs might start to encompass more of the experimentation. Nikhita alluded to, you can never encompass everything, but I think thankfully the science itself is going to lend itself towards being able to incorporate more of those in the cloud lab scenario.
All right, so Nikhita, a question back to you that we're going to end with because there's only like 3-4 minutes left. It has to do with company building and go-to-market strategy. Early in Artificial's journey, we were talking about a robotic fleet management platform, because the world needs something like that. But that's kind of very, Bob doesn't go out and, where can I buy a fleet management robotic platform? He buys a solution for his lab. So in that journey, so what Artificial is now doing is a lot of lab automation system. But does this eventually turn into a more generic platform where Artificial in some point in the future can actually move to other markets and reuse this platform? Are you thinking about that at this point?
Yeah, so I think anyone who's built a company and is building a company knows that hyper-focus is your number one kind of goal, right? It's limited resources operating under constraints. So I think right now we're focused on lab automation, but I think a lot of the principles you saw in the work that I was showing, like the digital twins and the natural language intent, right? I think a lot of those paradigms are really reusable across domains. It's certainly been my experience having seen robots in education and disaster relief and aging, where you always need to be able to encode intent and you always need to understand the physical world. And then you need to bridge how people interact with these systems. And so for us, we really thought about being intentional in the way that we design and create these tools so that it's something that can transcend just the domain itself with the, as if you add in the 20% that you need in order to specify it for a new space. So I think that is our hope and intent that we will get there. And we certainly built with it from the beginning. But right now we're pretty excited and happy to stay in the scientific lab space.
Absolutely, yeah. All right, well, we have 1 minute left. I want to thank our amazing guests, Nikhita and Bob, for your time and really, really, really good insights. I learned a lot. Hopefully our audience did as well. And we love having everybody back. Our next Robochat will be in September right now. And we're hoping to do it live with live audience in our conference room at Playground with a live stream. The topic will be related to ML ops and productizing AI. So it's going to be relevant to every one of our portfolio companies and everybody in the robotic space. So I really look forward to that one. And that's it for today. Nikhita, Bob, thank you.
Thank you for having us.
Thank you.
All right.